Well, welcome human biology class. Second to last week, we're going to be dealing with muscles today. We're going to be dealing with skeletal muscles. And this is an image from your textbook right here. Skeletal muscles are also called striated muscles. And you can see the stripedness that is apparent. If we had a microscopic slide like I would have in lab to look at, that stripedness is not always as pronounced as you see it here. It's there, but it's sort of a mystical looking appearance to the fiber. We've already talked about smooth muscle a bit. That was the muscle, for instance, along the digestive tract, causes peristalsis. You don't think about smooth muscle contraction. I'm thinking about making it work. We've also talked about cardiac muscle, layers of the heart muscle, the muscle that makes your heart beat, and it's a little bit unique. It's more branched if you look closer at it than is skeletal muscle. Skeletal muscle, this muscle we're going to be dealing with, is very uniform, very geometric, if you will. There's these very pronounced stripes in it. It is attached, if you look at your notes, to the skeleton by tendons. Okay, so if we look at images from your textbook, this is the one that I wanted to start with. Or go to second, I guess. So here's a bone, in this case the radius, one of the bones of your forearm. And remember, the bone is covered by periosteum. It's that tough, durable covering on bone. And if you ever go to the store and buy meat with a bone in it, I dare you to excise the bone out of it, and you will actually be able to see that periosteum. It's a very tough covering on the bone. It covers the whole bone, and tendons arise from that. And the point is that, again, muscles aren't just attached to a particular spot on the bone. They arise from that spot, but they're connected to this covering that covers the whole bone. Sorry, that brief timeout was for a pretty gratifying sneeze. Anyway, so we've got the periosteum, which gives rise to, in this case, tendons. It could also be ligaments, which attach bone to bone. But the tendons then become one with muscle. Okay, so the connection between muscle and bone, the point is, the connection between muscle and bone is much more intimate than this image of just sort of a muscle superglued to a spot on the bone. They are integral. They're one attached to the other. And I want you to think about how incredible your skeletal and muscular system is. I'm going to have you go right now to this page. There's a link. I want you to pause the video, and you'll notice this is about 3 minutes and 50 seconds into this video clip. You can watch the whole thing if you want, but I want you to really key on this last part. This gentleman is Brian Shaw. He's a Coloradan. He is huge. He's 6'8", and he is a deadlifting champion. He is not currently a record holder, but he has been many times in the past. Deadlifting is unique. There are several different types of rules to it that create different classes. But the bottom line is here, he is going to attempt to lift 1,128 pounds off the ground. He just has to get that off the ground. But remember, dude's 6'8". Okay, so I'm going to pause the video here for just a second, and I want you to look at that video clip. So hopefully you got a chance to look at that, and hopefully you're amazed by that. If you look at these gentlemen when they do this, it is not uncommon for them to start bleeding fairly profusely from the nose, given the extreme amounts of pressure and weight that goes on all parts of the body. But I want to just key on muscles and bones. You know, I should mention also, as he's lifting this, think of all. If I go ahead here just a bit to where he's actually lifting this, I hope YouTube police don't give me too much trouble here. Think of the pressure on his abdominal cavity as his muscles here push on his abdominal organs and try to push them through those inguinal canals that go south off the bottom of the abdominal cavity. But if you consider what's going on in his body, this is the musculature in the bones. But look at the bones. All that weight is being lifted and supported by little joints here, one on this side, one on the other. Or if we go down here to the lower leg, I'm going to go zoom in on that lower leg. all that 1,100 pounds and change is being supported by this spot right here, two spots, this one and the one on the other side. Just think of how incredibly durable and tough your bones are to be able to do that. And his muscles, and that's kind of the key point that we're thinking about here, is the muscular system that allows for Brian Shaw to lift that much weight. That's absolutely incredible. Well, if we look at our muscles, then we're again talking about the just the skeletal muscles, just the striped ones. Those are voluntary. So Brian Shaw was deciding to get his muscles to work to do what you saw. They can only pull. So muscle fibers only pull. So if you think about that, you're walking down the street. Well, how is it that some people can push then? Everybody can push, okay? So right here, if we look at the arm, so forearm and arm, here's the bicep, and so these muscles shorten. They pull when they are flexed, and that causes the forearm to be pulled towards the shoulder. It flexes it. You say, well, how then does somebody push? Well, the triceps right here, when they shorten, they move the forearm away from the body. So always muscles only shorten. That was redundant, but it was kind of intentional. Always muscles only shorten. And you'll never see a muscle lengthen to get a body movement to happen. So if we look then at muscles, their structure, we're going to zoom in on a particular part here. Again, here's that bone. This case looks like the femur, with a tendon arising out of it right here. And here's a muscle group. And you'll notice that the muscle group, I'm going to zoom in on this a bit. Maybe I am. Yeah, there we are. So if we zoom in on this, the muscle is divided. And there are these little packets, and if you look at a cut of meat sometimes before you eat, I know it's kind of a little bit harsh to think about, but we are set up just like cows and whatever else, pork, whatever else you eat. You can see there are different bundles in there, and they're separated by what we would call a fascia, a membrane that creates different bundles. That allows for this tendon here, again, not just to be connected around the outside of the muscle, but all through the muscle. We see those little connections that hold one part of the muscle to the other and then to the bone. And so if we zoom in on this, we can see here the basic functional unit of the muscle, that being the myofibril. Some might argue that it's the sarcomere. We'll get to that in a second. Your textbook has a bit of a misstatement. I don't want you to think about this muscle fiber or the myofibril, that many of which make up a muscle fiber. None of those is in and of itself a cell. As you developed, individual muscle cells fused to form muscle fibers. So there's no longer individual muscle cells here, even though there are the same parts, the nucleus, the endoplasmic reticulum, and those mitochondria, those features. There's not an individual muscle cell. And in fact, those individual cell parts have been given new names. Here we have sarco prefix on things, and anytime you see that sarco, before we had endoplasmic reticulum. Well, here we have sarcoplasmic reticulum. Sarco refers to muscle, so when you see that prefix sarco, it has to do with muscle. Okay, so the myofibrils then, if we look closer at them, we're going to see they're arranged specifically. So here we take this myofibril and we zoom in on it. This is an actual electron micrograph of that myofibril. Okay, and you can see layers or stripes in there, and this is some of what creates the stripedness that we see in muscle fibers, in skeletal muscle fibers. Well, they're there, those stripes are there, and those different areas are there because of the arrangement of different proteins. Actin and myosin are the two main proteins. Myosin is referred to as the thick fiber or filament, and actin the thin And it is the behavior of those, the way those interact with each other that cause muscles to contract. So we would then look at these lines here, these Z lines. So here's a Z line. Well, you can see it gets its name from this zigzag back and forth. It's not a perfect Z, but that's why they call it the Z line. So from one Z line to the other Z line, we would call this the sarcomere. And this is the part of the muscle fiber that actually shrinks as it contracts. Okay, so Z lines actually get closer together as the muscle contracts. There are a couple of other important parts. There is this membrane that covers the muscle fiber, and that is called the sarcolemma. Okay, that's important. And extending down from the sarcolemma, it actually has protrusions, they're colored green here, that extend down into the muscle fiber, and they're called T-tubules. And the T-tubules come in close contact with, they sidle right up next to these blue tubes here, which is the sarcoplasmic reticulum, analogous to the endoplasmic reticulum. Packages and processes proteins in the regular cell. Well, similar function here. But there's another key thing here. There are closely associated here little calcium storage packets that are going to play a key role in muscle function. So in the sarcoplasmic reticula, very close to where the T-tubules touch them, there's some relationship there that's currently being understood. We know that those sarcoplasmic reticulum have these little calcium storage pouches that are going to help muscle contraction happen. So calcium and ATP are the two main things that cause muscles to contract. Yeah, you got to have both of them. Remember how important I've been saying calcium is? Well, here you have it. It's important in bones. Calcium is important in heartbeat. Calcium is important in blood clotting. Calcium also is important in muscle function, muscle contraction. It won't happen without calcium. Okay, we're going to switch to another slide, and I've kind of put another spin on this in your notes. And I'm doing that intentionally because I think what's listed here in this step 1, 2, 3, and 4 is a little bit confusing sometimes. So really what we're doing here is we're going to look at what happens right here in the sarcomere when your body tells a muscle to contract. Okay, and if you look in your notes, there I have 6 steps. In your textbook there are 4. I've added some to them, and this is a real nice segue into the nervous system too. We'll refer back to this when we get to the nervous system. First of all, I want you to notice here. So right here, this would be a nerve, a neuron, okay, and this would be a muscle. So right here is the junction between a neuron, a nerve, and a muscle. So this neuron is going to carry, this nerve is going to carry the message from the brain, a message from the brain or spinal cord. Not all messages arise from the brain, some come just from the spinal cord, to this muscle to do something, to react to the environment, to do fight or flight, if you will, or whatever, okay? So to carry that message from the nerve to the muscle, there's a molecule that your body has called a neurotransmitter. In this case, it's called acetylcholine, right here, ACH for short. Acetylcholine, now interestingly enough, it's carried in little packets right here, right at the end of the neuron. So if we look here, so here's a bigger picture. Here's that nerve, okay? I'm using nerve and neuron kind of interchangeably here. The more correct term would be neuron. More common term is nerve. And so since I haven't parsed that out, I'm going to use them. Maybe I'll just try to stick with nerve. Okay, so here's the end of a nerve where it contacts a muscle. This is probably a muscle group. Okay, so if we look closer at that, there's this synaptic bulb. This is the end of the nerve right here. And then we're going to zoom in on that a little closer right here. And this is where the rubber meets the road. These little acetylcholine packets are held near the end, near this junction, and they're ready for use. Guess what, folks? It's calcium that causes these to move or to be moved to the edge right here so that they can be used. Once again, calcium is in the act is important. So acetylcholine goes to here, a message comes along, muscle do something, so the acetylcholine gets dumped into this space. It's called the synapse, or the space, or the cleft between nerve and muscle. And there are receptors on the muscle side that then receive the acetylcholine, and as soon as they're received, that's the stimulus that the muscle needs to contract. Okay, that causes calcium to be released. I'm going to go to another image right here. Okay, not that one, not that one. I got to go to another one here. Sorry, folks. Okay, so right here, if I go back to this one, so we're looking right here now. This picture right here. So we're looking at actin and myosin. Okay. So if I go to this image, so here we're looking at myosin and actin. So this is an actin molecule. So calcium. So that the impulse comes, tells the muscle to contract. The muscle, in response to that, the pouches along the sarcoplasmic reticulum dump calcium into the system. Calcium comes along to the actin molecule, and it bonds to the actin molecule, and it changes its shape. Okay, so that's step number 2. Then step number 3 in your notes, ATP is hydrolyzed to ADP and P. So ATP gets a phosphate cleaved off it. That cocks the myosin head. So this myosin, this is like shaped like a golf club. So if you can picture somebody's golf club bag, and the heads were here and the shafts were here, it kind of looks like mine. I played golf for the first time here last week, and it went pretty well, but sometimes I wanted to take the shafts of those golf clubs and twist them all together like this. Anyway, so what happens when ATP goes to ADP and P, energy is burned, and that cocks the myosin head. So this head of the golf club rotates back, almost like the hammer of a gun, if you will. And then we go here. Once that happens, this head, boom, it bonds to the myosin head. That bonding site is available because calcium came along and opened it up. Okay, boom, it locks to it. So the hammer's cocked, bond is made, myosin to actin. And then if you look number 5 here in the notes, number 3 here, that head flexes. Look at right here. That's called the power stroke. Boom, it flexes, and so the myosin goes from here to here, and it moves that actin molecule and causes the muscles to contract. Along comes another ATP molecule. It attaches to the myosin. That causes it to detach from the actin molecule, and the whole process can start over again. That's a bunch of words and a still image. At this point, it might be a good time to go back to Moodle, and I'm going to put a link there to a short video that puts some animation on this. It will give you, again, a lot more jargon, but try to keep the big picture in mind. ADP or ATP binds to myosin, cocks that myosin head. Okay, that myosin head bonds to actin. The myosin head flexes and moves the actin. New ATP comes along, detaches the myosin head. Cycle starts all over again. Okay, so take a minute, maybe look at that video and see what you think. Key thing here is, if you look at your notes, this is the next line. If this bond occurs and this flexing happens, it's all or none. There's no halfway point here. If this head flexes, it flexes the whole way. You say, well then, how can I decide to, like for instance, itch my nose? If I reach up to scratch the end of my nose, how do I keep from socking myself in the face, knocking myself out? If it's all or none, if that response has to go all the way or not at all, how do I have what we call fine motor skill? Well, the deal is, folks, that when a muscle fiber fires, that's true. It's all or none for the actin and myosin in a sarcomere here. But not all muscle fibers in the whole muscle group are going to fire at the same time. You have the ability to regulate that. So only a few fibers can fire and give you fine motor skill, motor control that you can see. Like if you think about the vocal cord surgery that you were witnessing, the person was manipulating a device through a microscope. Think of the fine motor skill that would be required for that to happen. All right. A couple of ailments that occur because of, actually I want to go back to that one, because of various issues that will arise. If I go back to here, we're going to talk about muscular dystrophy. And there are a couple of video clips that I'm going to have you watch, but let me explain the disease first. The dystrophin gene creates a dystrophin protein that attaches the sarcolemma to the myofibril. And if you think about it, there's going to be no contraction of the whole muscle group unless this myofibril, which is contracting, is somehow connected to the sarcolemma. Otherwise, this is just going to be moving back and forth inside the membrane and nothing else is going to happen. So there has to be a connection between this myofibril and this membrane, and that is the dystrophin protein. And it is integral to muscle function. It is also the second largest gene in the entire human genome. If you remember, genes are on average about 5,000 base pairs long. Cytosine, guanine, adenine, thymine, remember those bases. The dystrophin gene is 2.5 million base pairs long. It's huge. And that means there's lots of potential for free radicals, for instance, to bomb around and damage it. And so if it gets damaged, that's a problem. Now, the dystrophin gene is on the X chromosome. Here's any cell in your body will have 23 pairs of chromosomes. Okay, this one here is typically from a white blood cell, and they're lit up, colored specially, so you can see the differences in them. So there's a pair of number ones, a pair of number twos, a pair of number threes, so on and so forth. You got one from mom, one from dad. These chromosomes are the same size. Number twos are the same size and same shape from both mom and dad. This connection point is at the same spot. It carries genes for the same trait from both mom and dad. It just might not be the same gene. Down here on the X chromosome, which you got from mom, there is a gene that's where the dystrophin gene is. And now you see, if it gets damaged, that's going to result in a malformed dystrophin protein. And those misshapen dystrophin proteins can do a lot of things. Among them is they can block the calcium channel. They can cause a dysfunction of the whole system. Bottom line is, if that dystrophin protein isn't shaped right, it's not going to function right, and the person's myofibrils and muscle fibers will degenerate and go away as a result of that. And if you look at the video clips, Duchenne timeline, it's really heart-wrenching to see this. Now, this happens in boys, almost exclusively in boys. Not totally, but almost. Now, this is a genotype, the chromosomes from a male. There's an X and a Y. If it was a female, there would be two X's. The reason this is so common in men is because we have no other X chromosome. We only have our dystrophin gene from this one chromosome. In women, you have another dystrophin gene, and your body, if you get a problematic dystrophin gene on one X chromosome and a good one on another, your body may just choose to not express that one. It can handle that situation. But you see, men only have one X chromosome, and so this is predominantly a problem of young boys, and you'll see that on the Duchenne timeline, and it really is heart-wrenching. But I want to encourage you with one of the most breathtaking pieces of biotechnology applied to the human condition. That is CRISPR-Cas9, and that is the next clip that I'll have for you on Moodle, and it should be encouraging. And so that's all about what goes on when this sarcolemma is not attached well to the myofibril, and problems arise. Another one listed in your notes is rigor mortis. When a person dies, they're no longer producing ATP pretty quickly. And without ATP, you're not going to have the myosin detaching from the actin. Remember, in step 4 here, or step 6 in my notes, that's what happens. ATP comes along, it binds to the myosin head, and that causes it to detach from the actin molecule. Without an ability to produce ATP, as happens after death, that won't happen. And so the myosin stays bound to the actin, and the person's body remains rigid. Now, it doesn't remain rigid forever. Rigor mortis goes away within a day. And so the degree of rigor that rigor mortis that a dead body has will help a coroner determine the time of death. And as I say, they won't remain rigid forever. Another one is botulism. This is a toxin produced by a bacterium, Clostridium botulinum. Have you ever heard, when you go to the grocery store, don't buy a can of anything that's bulged? Well, that's true. The reason is Clostridium botulinum is a bacterium that is anaerobic. It grows without oxygen. And so if it ends up in that can, it can create CO2 and create that bulge and be sort of a giveaway that it's there. Well, what happens? Well, botulism, that toxin, prevents acetylcholine release at the muscle. So it's paralyzes that situation. So it would block this whole system of acetylcholine release and be a big problem, okay? Well, the deal is, some people, though, take that toxin from botulism and inject it into their own bodies. Your textbook has this image. There are actually even parties. They're called Botox parties, if you've heard of them. And what's in this syringe? Well, botulism toxin. So this lady's having this injected into her scalp, into her forehead here, to paralyze those muscles. Well, why, you might ask? Well, it's those muscles that often give rise to the wrinkles and crow's feet that we associate with aging. And this is right from the Botox main page, and here frown lines, crow's feet, okay, and here we go, crow's feet right here at the edge of the eyes, okay, not bad there, improved there because the muscles have been paralyzed basically, and here's frown lines. She's trying to frown here, but those muscles are paralyzed. She can't frown real good. Yeah, that's kind of interesting. But yeah, that's one of the uses for this particular toxin that people have taken from something that was bad and turned it into something that is fairly useful. Okay, last pieces today. ATP stored or in this clip. ATP, if you look at your notes under botulism, ATP stored or built in muscles. In your textbook, there's a very nice image of sort of 3 ways ATP is produced. Creatine phosphate, okay, those of you using that for a supplement, that's the purpose of it, to create more ATP so the muscles can work better and longer. That's an anaerobic, means without oxygen. Again, anaerobic, without oxygen. Fermentation, glycogen is converted right to 2 ATPs in glycolysis. No oxygen needed, but it's, again, fairly inefficient. You only produce 2 ATPs for every glucose molecule. And then there's aerobic, glycogen or fatty acids. Remember, both of those, either of those can be used to produce ATPs, but the key is here, there is oxygen. On the mitochondria, produces, up to around 34 ATPs with this one. So that's happening in your muscle. Your resting muscle is producing ATP and creatine phosphate. But during work, if you look down there under creatine phosphate, what's in your muscles, this is just what's in your muscles, the ATP that's stored up only lasts about 8 seconds. Creatine phosphate lasts about 22 seconds. Bottom line is, what's in your muscles gets used up in about 30 seconds. So your body has to get more ATP. And so if oxygen is present, it's going to do a good job of it, like you see in the images here. If not, no oxygen present, it's going to utilize one of these two pathways. The bottom line is, at some point, you're going to run out of energy. And I've got another clip here that I'm going to add of a lady doing a marathon, a Kenyan runner, really, and she was leading the race, and her body just quit. And so I'm going to add that clip at this point to Moodle, and you can see what that looks like. So what's going on is, at some point, you can push your body so far, but at some point, the hydrogen that is created as ATP goes to ADP, acidifies the muscle environment, and performance goes down and cramps ensue. Cramps are incompletely understood. Nighttime leg cramps, it's believed occur because of that laying in that prone position, maybe compression of nerves, not completely understood. Muscle cramps of any sort, we've all had them. They are believed to have something to do with dehydration and a swelling of that environment inside the muscle, and basically the calcium pump gets short-circuited, and cramping ensues. Again, not completely understood, but it does definitely seem to be related to dehydration in the muscle environment. Okay, so I'm going to stop there today, and I have some clips for you to look at. Make sure you avail yourselves of them, and have a good day.